Thank you for coming by this event. I appreciate your time and stop by our booth and talk with us. And I hope you have enjoyed the wonderful scientific program from ASSG, including the cool technology and the cool weather as well, right? So yeah, that is the year for Colorado, right? So the locations, the time matters. That's why you receive the snows. And ready or not, here you go. And that is also true for many, many things, right? So the locations and the time matters. So when you go shop a car, you would like, the manufacturing to properly assemble every single part into the car that he can drive with no worry, right? So that's what you expect it to. And when we talk about human health, that is the same thing. We will hope our different system in our body can function properly. There's nothing goes wrong with one system to another. Remember, we come to become a human. It's not exactly from the status what we have now. It comes from the long process. It starts from one cell and then goes to divide, and then they're kind of temporally and spatially organized together. That is how they build the human bodies. And when things goes wrong, and you can imagine those time and spatial also matters, that's why we need to think about this spatial and locations and temporal information as well. So of course, for the secret of life in the past several decades, scientists have spent a lot of efforts to understand the secret of life. So right now you are all scientists. There is no more saying from me. We know the central dogma, right? So the government of the life. So from the substance-wise, we have the DNA that is governing every single phenomenon in our body. And then this DNA has to go to the RNA and the protein in order to have our bodies to function properly. So that is the technology advancement makes the scientists to do these things in the high-throughput way and in the very sophisticated way to do these studies. And for a long time, from the cell concept about the life, and since then, the technology advancement, as you can see, we can sequence in the DNA, we can sequence in RNA, and we can sequence it in the high-throughput way. We can sequence it in the very fast and economic ways. And in the past few years, and we can also put all this information in space, and we can understand where they come from, how they interact with their neighbors, and what kind of mechanism they come together to drive that kind of specific physiology functions. So Complete Genomics, as well as Stereo-seq, come up with a fantastic way to do this. And we believe this is still the most advanced technology in the field today. We use the in situ capture technology, as you can see from the left-hand side. We manufacture the DNBSEQ chip, silicon chip. On this chip, we placed millions and millions of the oligos that can grab the RNA based on two concepts. If you have a fresh frozen samples, your RNA still stays in the native structures, and we can use the poly-T hybridization and grab mRNA from the tissue. And if you have the fixed samples, or your sample has some sort of the degradation, and you processed with FAPE, we can still come with the solutions that offers this high resolution and species agnostic ways to do so. And we have the randomers to grab the total RNA, not only just mRNA, but total RNA, even microbial. So what you can do from here, you purchase the products from us, and you just place the tissue, and then you can do everything. And when it was in one run, you will end up to know every single cells on your tissue. And you can profile the whole transcriptomic, and just to see what kind of mechanism may be involved in your specific questions. And that technology made the cover for the Cell paper in 2022. Not only just design those products that you can work with your tissue, but also the scientists from the company, they come up with the total solutions. They provide you the software for free, and you can download and you can install on your computer, and also we provide support for you to run this software, and then end up you can integrate the different layers of the information, the images, the molecules, and also about the different ways to do so. You can group them in the macaque way. We can call bean size. You can choose a different size of bean, and you can do that. Or if you are really interested about the individual cells, you can, based on the image you captured under a microscope, and you based on kind of AI-based algorithms, or use the immunofluorescence membrane proteins, you can do the accurate cell segmentation. So on the right-hand side, you see that is what you can do from your data set. After you run all this workflow, and then you're going to understand the different populations in the right locations and how they're performing their function. So as you can see, we believe this is the advanced technology, and the scientific community acknowledges that as well. Within the short 2 years, we have more than 80 peer-reviewed publications on the high-tier journals. And this becomes becomes established a protocol for around the world for over 100 labs that they can provide their services to the scientists as well. So I'm going to talk more about these technology features. So for Stereo-seq, we are very proud to present the 3 major functions. The first one, we believe, based on the high resolution, 0.5 micron, right? So based on the high resolution, we can really do the true single cell segmentation. That empowers you to profile the cell, not just the area, that you can do this analyze. And also we provide the big chip size, as you can see from the table, from the very smaller one, 0.5 centimeter to square, to up to 13 by 13 centimeter square. So with this size increase, we're not sacrificing with the resolution. As you can imagine from other technology, if you give the bigger ROI or capture area, you have to sacrifice about the resolution or the sensitivity because you don't have that much time to image everything, right? So for us, we have that flexibility. You don't have to sacrifice with the resolution. Another thing is by design, this technology is designed for working with any organisms. So this is a species agnostic. It's not designed a probe to target the human and mouse. So as long as you have a well-annotated genome for that species, we can work with it. So we can just create the reference genome, and you can do the one run of the sequencing. You will know a whole transcriptomics for every single cell. We believe that is the true power of this technology. And these slides, we just kind of highlight the comparison between the different technologies, right? So most of those already know many of this technology. As you can compare among this, and according to the different categories, we believe our high-resolution true single-cell segmentation with the species-agnostic features can stand out from the current technology landscape. Yeah, this is another, we just want to talk more. This is a real example, from the mouse brain one by one, and you can all the way to the larger chip to profile the human brain even. And speaking of the species agnostic, the company and our users have tested different sample type across the different species. Right, so this is just a lot of exhausted list. This is just maybe from last year. So this list is becoming longer and longer. In order to run this assay, it's very simple. You start from your sections, you prepare your sections, and you do the QC, and you purchase the kit. And then you can do this kind of histology work and do the imaging work. It's just 5 minutes staining if you don't do the antibody staining. And then you do a capture image. And you use all these reagents that we provide. You can make a library. So after you make the library, you can send to the very powerful DNBSEQ sequencer to do the sequencing. We have a different throughput sequencer available, which is fantastic. You have everything here, and you can do the sequencing. And then after you get the FASTQ, and we have the free software you can download and you can run, and we have the bioinformatics people to train you how to use it. And you can analyze your data and visualize, crop it and put it in your presentation, grant a proposal, or grant report, or even publications. So that is the technology. So how can this technology support your big studies? And you can imagine there has many, many research fields that you can work on. And here I want to spend a little bit of time to talk about several exemplary studies that people have used this technology to help them to understand the big questions. As a vendor, we are different vendors than the ordinary vendors. When we provide the products, we're not only wants to sell you the product, we really want to help you to drive the big ideas, to help you to have the tools that you can just focus on scientific questions. And we provide all this support for you to think more, think big, and we can provide the support and products and support. So for the first example, I want to say, think about the Stereo-seq, and we use technology to support the neuroscience. When we talk about neuroscience, this is a big field. There are many aspects you can ask the questions from. For example, we talk about Atlas, right? This is the foundations for people to work in the neuroscience field, what their cells, how they're generated, right? And then you will see how they're developed, how this neuron system evolved across the species. And also, collectome. The neurons are not individual cells. Their locations and collection matters. And also the disease, of course. And then eventually, we're trying to decoding the neuron system, the very, very complex system in the human body. So I will just use one of the examples to cover all this kind of field. Here in the past year, we published the macaque brain atlas. So we used 160 big sections, and we profiled 100 millions of cells across the anterior to the posterior area of the macaque brain. And from that studies, you can create a 3D reconstruction about the different cells, how they're organized in the cortex columns. And from that study, they found the specific cell type in the layer 4 that is primate related. And they found the mechanism structure, molecular signature about that specific populations of the cells. So this is an example that people using this technology to provide the foundational that every single scientist in the world, they can download this data and they can run and analyze them for their own questions. So this is another field talking about the development, right? The researcher using this technology and profiled the cells, their proliferation, their molecular signature, and how they're organized the human brain during the development. And as you can see, all this kind of features has been profiled among this kind of different spatial axis as well as the time axis. This is another studies. People want to understand how the human brain becomes a human brain, right? So all this brain starts from one cells and from this very simple system, and how this kind of simple system becomes a complex, the human brain system. So these studies, they compare the different species, the mouse, marmoset, and macaque, and then they analyze the specific area in the cerebellum. And what they found here is there has a primate unique Purkinje cells. And if you can interested about these details, you can read more about this paper. And they found the molecular signatures among the primate. And that's how those cells becomes primate-specific cells. And this is another study in the area that people understand about the collectome, right? So people really want to understand how the cells are connected with others. And then in the past, people can only use the electron microscopy or light microscopy to see one cells, how they're connected with others. This is the physical connections. There is no molecular connections among the different cells in different area, because that's just simply impossible to do so. With Stereo-seq, people combine the Stereo-seq technology with the line microscope profiling, so they can really understand how the hippocampus neurons and connect with the rest of the brain. This is just one region of the example. You can think about there has so many nucleus, right, for the Parkinson's disease, for the Alzheimer's disease, for the ALS. So all this kind of disease you can focus on, you can really understand their connectome and also understand their molecular signatures. It is possible. The tools are here. So talking about the disease, this is another example. People want to say, hey, the metabolic disease is become very problematic or wide. So people are building more knowledge based on the previous findings and say what other cells might be involved, because we understand all this technology by now, but we still cannot cure this kind of metabolism disease. We need to find new mechanisms. And from these studies, they create a model about obesity and type 2 diabetes, and they compare with the different conditions and treatment. What they found out is there has another neurons called PVN, that area in the hypothalamus, So they have a particular signature in the diabetes compared to the obesity, right? So all these kind of molecules might be involved in this disease process. This is just one of the examples. Of course, Dr. Sen, our second speakers, will talk more about another fantastic models using the Stereo-seq, and you will hear more about from that about the Alzheimer's disease studies. And then we have a user in the US, and they shared their results in the SFN conference. What they really want to figure out using our Stereo-seq technology is they really want to figure out the function of the brain and how they're connected with the signature of the molecular transcriptomic factors. So stay tuned. Their paper will be, hopefully come this year. Another example I want to share is Stereo-seq is actually changes the research paradigm. In the past, or you write in the proposal, and the NIH always asks you hypothesis-driven. You got to tell me your hypothesis based on the literature, based on your preliminary data, and then you tell us what you want to do and alternate way to test it, right? So that is the normal path you are going to do. So here with this Stereo-seq and other technology, we change the paradigm. You don't have to have that first hypothesis in the first place. And we're going to generate the humongous big data set that for you to dive in and dig deep into it, and you can analyze the different mechanism, different signaling pathway in the different conditions, response to the treatment or not response to treatment, right? All this, you can generate the data without having any preset hypothesis. And from there, you can dive in and look at the different mechanism. And here is the example how the Stereo-seq generates those data, and you can see that provides you that foundation. They just look at those data and see if something interests you. And I want to use this as a free example. So why we're using this paradigm and help the researcher to build their research study. And this is a study that talk about the liver cancer, right? So across the cohort studies, the researcher found there has the molecules are increased. And whether that's really involved in this process or not, and the design experiment with the histology, immunofluorescence, Stereo-seq, and they look at this like in the high-throughput way. And they found a lot of genes upregulate, downregulate in the invasion zone, is that where their normal tissue and the tumor tissues comes together. That's a unique invasion zone, and they compared this unique zone with the other region of the cells, and they found that there has a critical chemical, SSA4. And then in order to test that, they locked down that protein and generate the animal models and test it. And they found the tumor invasion has been slowed down. So that means this Stereo-seq helped them to find the molecules and then generate a hypothesis and design an experiment to test it and see the results. So here is another user case from MD Anderson. They're working on the ovarian cancer. Of course, there has a lot of findings about ovarian cancer, but this study, what unique about this is they really combine the Stereo-seq with other spatial technology as well. So they can create the 3D multi-module omics studies. They provide the framework for any other researcher to do it. So based on this study, this is a very preliminary analysis. As you can see, some of these molecules already known, some are not known, so it's new for the researcher to dive in and find the role of those molecules in the progression of the ovarian cancer. So, of course, aging is very predominant in this world, right? Modern world, when we become more aging people in the world. So we want to understand the fundamental mechanism about the aging. So the aging is not only the brain aging. All other tissues, organs are aging as well. Are they aging in the same pace? Are they using the same mechanism to become aging? So we do have like different studies in the past talk about the different organs and different tissue, right? So this study really put this together as a united effort. This study about the different time, right, from 2 months to 25 months in the different age of the animals, and they studied 9 organs across the body. And they understand how the synapse cells become a problem for that tissue, that organ, and for that individual. So here, I'm not going to details about their findings, but on this paper, they talk about genetically geographic features, and they found even more here. They're just giving other features about how the tissue or organ becomes aging, right? So they create a OSE, that kind of like an index to indicate the aging process. So as you can see in the young age, so those kind of tissues are well organized, the cell allocation, the cell identity remained, and they're using the example about the immune organs. As you can see, the B cell, T cells are well organized in the young age, but those structures becomes disrupted during the process of aging. And of course, they found there has the markers about this aging. So in the synapse cells, around that, there has an increased IgG expression. And this is not coexist, they're co-present, right? So they're co-present in that micro-leach. So this microenvironment, we can call it a leach, and then this microenvironment becomes problematic across the organ. And it's not the same for all the organs. This process is heterogeneous. So in this study, powered by Stereo-seq, and provide that tools that researcher can really think big, that people hasn't done, and they can do this in a high-throughput way, in the economic way, in the very sophisticated way to investigate those problems. With that, I will say, we're here and we're ready to support you to think big and support you with the big ideas and big questions. And what I want to share lastly is about this Spatial Accelerator Program. So the company together, we are going to support you with the kit and the sequencer reagent as well. We want you to think big, bring the big projects to us, and we will support you with not only just reagent kit, but also the support and also the other support that you can think of. And the more information you can find, let me just see if I can click here. Sorry, no internet. But you can search like Complete Genomics Accelerator, and you will get that program. That's all. Thank you so much.